Bismillahirrahmanirrahim. Assalamu alaikum. I'm Sadia Sarfar along with my group member Ali Yasmin from BS Chemistry 4th semester Fatima Jinnah Women University Rawalpindi. And our topic of discussion is the determination of metals and their oxides by different types of titrations including complexometric, precipitation, redox and acid-base titrations. I'll start with the determination of a single metal through complexometric titration. The first step is dilution of the sample solution to 100 milliliters in a conical flask. In second step, the buffer solution is added to it along with the indicator mixture, and in the last step, it is titrated with EDTA, ethylene diamine tetraacetic acid, till the end point is reached, which is normally a color change of the indicator added. The complexometric titrations can also be used for analysis of different metal alloys as well as for the separation of particular metal from mixtures of varied concentrations. The first alloy to be analyzed is brass, which consists of copper and zinc but in varied proportions. The modern brass consists of 67% copper and 33% zinc. However, these percentages may vary, which results in different kinds of this alloy. It's basically used for some decorative and mechanical purposes. This analysis is done through complexometric titrations, and the reagents used for this are the pieces of brass, 0.1 molar EDTA solution, the xylenol orange indicator shown here, and nitric acid solution. The first step is the preparation of 2 samples of the test solution by reacting the brass pieces with 5 milliliters of concentrated HNO3. This nitric acid is used for the dilution of the pieces of the brass which are added. One of these solutions is titrated with 0.1 molar EDTA, and xylenol orange indicator is used here. The equivalence volume for this titration is indicated by the color change from red to green. Now, prior to the titration of the second sample, sodium thiosulfate is added to it, which masks the copper ions. Now only the zinc ions are available for titration with EDTA, and the color change from red to yellow gives us the equivalence volume for these zinc ions. In the first titration, the total concentration of the cations, copper and zinc, is found out, and the complexes they are formed simultaneously. But in the second titration, the addition of that sodium thiosulfate masked the copper cations, so only zinc ions were available for titration. With the help of these 2 titrations, the concentrations of the copper and the zinc are determined. Similarly, chromal, an alloy which contains nickel, iron and chromium, can also be analyzed for the weight percent of these cations through complexometric titrations with EDTA as titrant and murixide indicator. This alloy has different types depending upon the concentration of its components. Alloys of aluminium, copper and zinc, they can also be analyzed through complexometric titrations. The complexometric titrations can also be used for determination of nickel in different samples, and here the nickel plating solution is analyzed next for the determination of the nickel concentration in it. This can be proceeded through either direct or back titrations. The reagents used for this titration are 0.01 molar EDTA solution, sample nickel solution, the murixide indicator, and a buffer solution for maintaining pH around 8. This chemical equation here shows that 1 mole of both of these reagents is used to give 1 mole of the product, the nickel EDTA complex. The first step in the procedure is dilution of the nickel solution in a conical flask up till 100 milliliters with the addition of distilled water. In second step, a pinch of indicator is added to it, and then it is slowly titrated with EDTA solution. The process is continued till the solution color changes from yellow to violet, which gives the end point of this titration. As it was shown by the chemical equation, the metal ion and the titrant, they are in 1:1 concentration. So the number of the moles of the nickel is exactly the number of the moles of the EDTA used. So in this way we can determine the concentration of the nickel in any sample solution. Other than these sample solutions, nickel can also be determined in its synthetic alloys with EDTA and murixide indicator in a buffer solution through complexometric titrations.
This is Alia Yasmin, and I am going to further continue this topic of application of titrations. The soil toxicity level is checked by determining the concentration of trace metals by complexometric titrations. It is run by using any of the three given complexes. We can use EDTA, DCTA or EGTA. The structure of these complexes are shown in this slide. The trace metals concentration of soil can also be determined by redox titration. In order to determine the concentration of copper, murixite indicator is used, which shows the change in color from orange to purple. For nickel, again murixite indicator is used, which changes its color from yellow to purple. For zinc, eriochrome black tea indicator is applied and color is changed from wine red to blue. Next application is determination of iron content of ore by redox titration. For this process, titrant solution is iron ore dissolved in HCl. Titrant is potassium dichromate. Indicator used for this process is diphenylamine sulfonic acid. Ferric oxide is our analyte in a given cell of an ore. For this process, ore is first dissolved in HCl. Then the oxidation state of analyte is pre-adjusted before the titration. Now titrate the given sample against the potassium dichromate and calculate the percentage of ferric oxide present in ore by calculating the volume of titrant used for the titration process. Now the purpose of pre-adjustment of analyte oxidation state is to maintain a single oxidation state within an analyte sample. And for this we use a device known as Jones reductor. A typical Jones reductor has a diameter of about 2 cm and holds a 40 to 50 cm column of amalgamated zinc. And amalgamated zinc is actually the zinc granules in a solution of mercury 2 chloride. This chemical equation is the representation of amalgamated zinc. You can also see the diagram of a reductor where a solid reductant is filled first and analyte solution is added above it. As you know, the reduction is the gain of electron. So here in this step, the oxidation state of iron gets changed from +3 to +2 by the gain of electron by using the reductor. In oxidation half reaction, iron changes from cuprate to cupric with the loss of electron, while in a reduction half reaction, the oxidation state of chromium is changed from +6 to +3 by the gain of electron. The last one is the net ionic balance equation, where iron is oxidized while chromium is reduced by the acceptance of electron from the iron. For performing mathematical calculations, first of all, the number of electrons and number of moles are conserved on both sides of the redox reaction. After that, multiple steps are performed and values are put into the formulas for calculating the percentages of iron oxide in a given sample of ore. Next application is spectrophotometric determination of zirconium by complexometric titration. First, use benzene as an EDTA solution for use as a gentle non-enzymatic cell dissociation reagent. Reduced spectrophotometer with the UV light is used. Now direct complexometric titration of zirconium with benzene takes place by using standard versinate solution of 0.0134 molar and add 5 grams of EDTA dissolved in water. In the next step, ignited zirconium dioxide is dissolved via potassium bisulfate fusion. In the last step, solution is titrated against the standard zirconium solution and the end point is the light yellow color.
That's all from our side. Thank you so much.